The overall goal of the following experiment is to quantify cellular invasion by the pathogen Listeria monocytogenes within the context of siRNA assays to discover novel cell functions required for bacterial infection. This is achieved by initially performing a cellular siRNA reverse transfection to inactivate specific host cell functions. As a second step, cells are inoculated with Listeria monocytogenes, which will differentially infect cells according to the cellular functions that have been affected by the siRNA treatment. Next, infected cells are processed by immunofluorescence in order to label the cellular nuclei, the actin cytoskeleton, and the bacterial protein InlC. Results are obtained that identify infected cells and distinguish them from non-infected cells based on microscopic analysis of the distribution of the InlC protein in individual cells.
The main advantage of this technique over existing methods like gentamicin invasion assays is that it can be used in a high-throughput manner.
So this method may help answer key questions in the cellular microbiology field by identifying novel signaling pathways required for the internalization of Listeria monocytogenes into host cells.
"72 hours before cellular infection, bring to room temperature a black 384 well microscopy cell culture plate containing 1.6 picomoles siRNA in 5 microliters of RNase-free water in each well.